Hello, everyone. My name is Monica Krzyzanowska, and I'm a medical oncologist in Toronto. Over the next 10 minutes or so, I'm going to discuss our pragmatic cluster randomized trial called AToM, which evaluated a complex intervention aimed at preventing emergency room visits and hospitalizations amongst women with early-stage breast cancer on chemotherapy. I'm going to review why we undertook this trial, the design, the results, and lessons learned. So let me start with what was the problem that we were trying to address with this particular trial. So most contemporary chemotherapy is delivered in ambulatory or so-called outpatient settings, which means that when patients experience toxicity from their treatment or complications from their cancer, they do so at home between visits to the cancer clinic. Population-based studies from across the globe suggest that emergency department visits and hospitalizations are very common during chemotherapy, especially in routine care settings. For example, in a prior study from Ontario, Canada, conducted by our study team, 43% of women diagnosed with early-stage breast cancer who received adjuvant chemotherapy experienced at least one emergency room visit or hospitalization during treatment, compared to 9% of non-cancer but age and comorbidity matched controls. It is not surprising, then, that there has been substantial interest in identifying ways to remotely monitor patients during systemic cancer therapy. Telephone or digitally enabled remote symptom monitoring during cancer treatment has been shown to improve patient outcomes and healthcare utilization in early phase and proof of concept studies, but large-scale evaluations of this type of approach were lacking. The overarching objective of our work was to evaluate the impact of a proactive telephone-based toxicity management approach on emergency room visits and hospitalizations in women with early-stage breast cancer receiving either adjuvant or neoadjuvant chemotherapy. We began with a pilot study, then moved on to a pragmatic cluster randomized trial following the successful pilot. Results of the pilot study are summarized here. Our aim was to evaluate the feasibility, acceptability, and preliminary effectiveness of this approach. Our target population was women receiving adjuvant or neoadjuvant chemotherapy for breast cancer at 2 cancer centers in Ontario, Canada. The pilot showed that the intervention was feasible with respect to recruitment as well as adherence with calls. It was also acceptable to both patients and providers and was associated with fewer emergency visits in the intervention patients relative to matched contemporary controls. We then moved on to a pragmatic cluster randomized trial with the same target population, where in the pilot we individually recruited patients and followed them over time. For the cluster randomized trial, we introduced the intervention at the participating centers as a process improvement. In addition to the intervention, both in the intervention and the control centers, there was a patient-reported outcome sub-study where at least 25 patients per center were recruited to complete questionnaires on several important patient-reported outcomes. This slide summarizes the intervention details. In the control centers, patients were provided with standard of care chemotherapy teaching at the start of treatment, and then a reactive model of care for chemotherapy side effects and toxicities, wherein they could call the clinic if they were experiencing some symptoms or toxicities they needed support for. In the intervention centers, patients received chemotherapy teaching as per standard of care, but they were also given a self-management booklet that focused on common toxicities experienced during breast cancer adjuvant chemotherapy, and also received proactive standardized telephone calls from symptom management nurses at 2 time points following each cycle of chemotherapy. For the PRO sub-study, we collected patient-reported outcomes at baseline, prior to cycle 2, and within 30 days of completing chemotherapy. For the subset of patients who switched their chemotherapy to a taxane halfway through, we also collected an extra time point for the PROs prior to the second cycle of taxane-based chemotherapy. With respect to sample size considerations, our statistician used 2 different simulation approaches using real-world data from Ontario to determine the optimal sample size. With approximately 73 women per center from 20 centers, we would achieve an 80% power to detect a 33% reduction in the number of emergency room visits and hospitalizations with a one-sided alpha of 2.5%. For the patient-reported sub-study, At least 25 patients per center would provide 80% power to detect the treatment effect size of 0.35 standard deviation, which is considered a small to medium effect size. Given that the different centers were different sizes, meaning different number of potentially eligible patients, the recruitment window for each center varied from 6 months for large centers to 1 year for medium and smaller centers. And how we defined a large center was to have at least 150 eligible patients per year based on historical data. Our primary outcome was the center-level mean number of emergency room visits per patient that occurred during chemotherapy, which was defined as day 1 to 30 days post last cycle of treatment. And this was determined using Ontario Healthcare administrative data. With respect to secondary outcomes for the PRO sub-study participants, we assessed the effect of the intervention on severity of toxicities, self-care, self-efficacy, quality of life, and coordination of care. We are also planning to do a cost-effectiveness analysis depending on the outcomes of the trial. I wanted to spend a little bit of time describing how we were planning to analyze the primary outcome, as it was done using administrative data, which is not typical. To identify eligible patients, we used the provincial chemotherapy billing data to identify all women with stage 1 to 3 breast cancer who started adjuvant or neoadjuvant chemotherapy during the recruitment period at the 20 participating centers. We then linked that cohort to provincially available data on emergency room visits and hospitalizations, and counted all the emergency room visits and hospitalizations from start of chemotherapy to the end of chemotherapy plus 30 days. We then compared in an intent to treat approach the center level mean number of emergency room visits and hospitalizations per patient at the center level between the intervention and the control arms. This slide summarizes the demographic and clinical characteristics of our cohort. Out of 2,158 patients, there were 2,158 patients that received chemotherapy during the trial period. Of these, 580 patients participated in the PRO sub-cohort. We saw no significant differences in the demographic or clinical characteristics between the two arms. With respect to the primary outcome, there was no difference in the center-level mean number of emergency room visits and hospitalizations per patient between the two arms. The mean number of emergency room visits and hospitalizations in the intervention arm was 0.91, whereas it was 0.94 in the control arm. This slide summarizes the toxicity data, which was based on the NCI Pro-CTCA instrument, a validated patient-reported outcome measure. There was less grade 3 toxicity reported in the intervention arm, with 48% of patients reporting at least 1 grade 3 toxicity, compared to 58% of patients reporting a grade 3 toxicity or greater in the control arm. The majority of these differences were in symptoms such as fatigue, aching joints, and aching muscles. We also saw a lower decline in the quality of life from baseline based on the FACT-B instrument, which is another validated measure, with a statistically significant lower decline on the trial outcome index, as well as the physical well-being subscales. We did not see statistically significant differences between arms in the other self-reported outcomes, including self-efficacy, anxiety, depression, coordination of care, or global quality of life as measured by the EQ-5D instrument. There are a number of unique aspects of our trial that warrant highlighting. First, with respect to the design considerations, the approach that we utilized to introduce the intervention in the centers as a process improvement was innovative. We also utilized existing administrative data to determine our primary outcome. This was possible because of the healthcare system organization in Ontario and the availability of provincial comprehensive database for the outcomes of interest. We appreciate that this may not be feasible in other healthcare jurisdictions. We did encounter some implementation challenges, despite the fact that we addressed most, if not all, of the implementation barriers identified in the pilot. Several centers had workflow issues related to identification and enrollment of eligible patients, which resulted in suboptimal penetration at some of the centers, ranging from a low of 50% to a high of 86%. Availability of nursing resources and training of nurses remained an issue, despite the fact that we provided dedicated financial support to hire nurses for the study. Additional limitations included the fact that we focused on a single disease entity, but we did include many different centers, including academic and community centers. We did not do any risk stratification for the patients, as there was a lack of accurate predictive models for acute care utilization during therapy at the time of our study. There were also some temporal changes in supportive care across Ontario during the study period, specifically for supporting patients experiencing toxicity during chemotherapy, which may have resulted in a co-intervention, especially in the control sites. Lastly, there were some potential ceiling effects that we saw for some of the secondary outcomes, such as the Stanford self-efficacy scale, with very high values at baseline. So in conclusion, in our 20-center cluster randomized trial, remote proactive telephone-based toxicity management during chemotherapy for breast cancer did not lead to fewer emergency room visits or hospitalizations, but was associated with fewer grade 3 toxicities and a smaller decline in quality of life. We were able to demonstrate the feasibility of using administrative data to evaluate healthcare utilization outcomes, such as emergency room visits and hospitalizations, in a large study. From an implementation science perspective, addressing implementation barriers identified in the pilot study of complex interventions may not be enough to overcome implementation challenges associated with scale-up and large-scale evaluations of the same intervention. And this warrants further research. Until then, the effects seen in individually randomized trials of complex interventions may not easily translate into population-level impact. So thank you very much for your attention, and I should mention that originally this presentation was made at the 2020 European Society of Medical Oncology Congress.